Hello dear students, welcome to the another video session of parasitology. In that we have started the chapters 5, that is the life cycle, pathogenicity and control measures of the various parasites. In that we have completed Plasmodium vivax. We are completing today Entamoeba histolytica. In that we have seen the systematic position, geographic distribution, all okay. We have seen its morphology, the various stages, nutrition. In last session we have seen its life cycle, okay. So its trophozoite stage, then get transferred into the pre-cystic stage, after that uninucleate stage, binucleate, and then quadranucleate, that is the mature cyst, okay. So we have seen the exhibiting the various stages in that the life cycle of Entamoeba histolytica, so that these incestation and the excistation, okay. So the incestation lies in between pre-cystic stage and the uninucleate stage, okay, and excistation lies between quadranucleate cyst stage and the trophozoite stage, okay. Here the quadranucleate cyst stage is uh, expelling from the intestine or the gut of a human and transferred by means of the feces, okay. And then it, when that cyst or quadranucleated cyst is going to take chance to get invade the new human being or the new host, okay. So in that, after the getting chance to get infested by means of to the new host by means of the uh, drinking water by carried out by some vectors, okay. In that intestinal situations, so these after this process known as the excistation is completed to get convert quadranucleated cyst to the trophozoite, okay. In that various modifications are there, so that modifications and the its treatment, its precautions we are going to see today, okay. In the excitation stage, that is stage in between the quadranucleated cyst to get convert into the trophozoite, okay. We have seen the various types of the ulcers, okay, that are the flash shaped ulcer is histopathology, okay. So we have seen its mode of infection and transmission by fecal oral route, and the process of excitation we have seen, okay. The, as is you see in this picture, that invasion, okay. After this invasion, the various types of the organs or the supporting organs to the digestive system get infected by means of this invasion, okay. And the process of excitation we have already seen, okay. So in this part today, that is part third of Entamoeba histolytica, and this will be the last part of Entamoeba histolytica as well. So we are going to see the pathology of Entamoeba histolytica, the some highlighted points, that are the incubation period in man varies a great deal depending upon the host resistance and the parasite, okay. So the incubation period, for we have mentioned in the last sessions that the incubation period for this species varies in between 10 to the 14 days or the 10 to the 15 days, okay. So that uh, gap in between, that varies the host resistant to the parasite, how that host is going to react after these get infested by means of the parasite, okay. So the generally it is a 4 to 5 days, okay, and that is symptoms of the disease appear in 4 to 5 days, okay. after the host being infected with the parasite, and total the uh, diagnostic stage will be the 10 to the 15 days, I said. Okay, so the incubation period varies from the 4 to the 5 days. Okay, so the pathological condition arising out the infections of Entamoeba histolytica is referred as a term named as the amoebiasis. Okay, so the amoebiasis is the term that's coined, or the scientific term, or the medical term, okay, has been defined by the World Health Organization as the condition of harboring these protozoan parasite Entamoeba histolytica with in or the without clinical manifestations. Okay, so the symptoms of the disease, however, appeared in only 10% of the infected individuals. Okay, so the 10% infected individuals are capable to see or the get some symptoms. Okay, so as you see in this picture, that will be this invasion part. Okay, invasion part. What is that part is the, okay, you can see the uh, amoeba which is carried out by means of the bloodstream. Okay, okay, and that is known as the invasion. It may you look like in the lungs, in the liver, okay, in the pancreas or in the brain. Okay, so that will be our invasion part. Okay, so how that some amoebic uh, going to get invade in these particular organs. Okay, so the effect on the host or the pathogenicity of these uh, particular parasite on the host, that will be, the first will be the amoebic dysentery. Okay, so the first after these getting invade in the our digestive system, that the that particular host is suffering from the amoebic dysentery. Okay, so the proteolytic enzyme secreted by this trophozoite stage of this particular parasite, which damages the mucous membranes of the intestine of that particular host. Okay, and these presence or and actively of these parasite in the wall of that large intestine cause amoebic dysentery, and these characterized by frequent passage of a stool gripping a palm. Okay, some cramp like you can see some, you can feel like some cramp. Okay, and you can feel like the gut is you can feel like to go to the toilet, but you are, if you are sitting there, but you are not uh, overcoming for that particular dysentery purpose. Okay, and the various cramps are there. Okay, the pain in these particular intestinal gut can occur. Okay, this is happen because the proteolytic enzyme are secreted by means of this trophozoite stage of this particular parasite, that is the Entamoeba histolytica. So second part will you suffer from, that will be the abscess formation in the various organs. But when this abscess formation is going to get in condition, that will be when that particular term, what we are going to see, that invasion occurs by means of the bloodstream, that when that parasite carried by the bloodstream into a liver, lungs and brain causes the metastatic and lesions, and the after that lesions formation over there from except from this intestine. Okay, so that lesions leads to uh, abscesses means pus formation. That lesion leads to the abscesses which may cause death in the severe injuries. Okay, so the infection of a liver called hepatic amoebasis. Okay, and the infection of the lungs called um, or the lungs amoebasis. Okay, so in the brain these parasite cause a cerebral amoebasis. Okay, so these are the some effect on the host or the pathogenicity of that particular parasite to the host. So the some parasitic adaptation, this will be the additive point to get better understanding, that will be the parasitic adaptations. First will be the morphological adaptations of that particular parasite, what that morphology is get changed when the infection occurs to the host. Okay, so the morphological adaptations, that are the parasite has lost its osmolarity organs. Okay, the osmolarity organs or the organelles are lost or they get degenerated. Okay, and as the host environment of that particular intestinal environment is isotonic in nature. Okay, so what happen if these osmoregulatory organelles are not get degenerated or not get lost? Okay, when that parasite is going to live in the water filled or the rich in the water of that particular in the intestine, then the osmosis is going to occur and that particular parasite is going to get increase its size. Okay, so that this simply that trophozoites are not going to get chance to get wandered into the intestine. Okay, so that the this locomotion will be lost by means of this particular parasitic stage, that is the trophozoic stage. Okay, when that's only single pseudopodium is enough for locomotion as it lives in the food rich environment. Okay, so that they are not going to locomote so deep or the so long. Okay, if say they are living into the food rich environment. Okay, so this isotonic nature of these intestine, they can lose their property of these locomotion, so that they are going to change in their selves. Okay, the parasite is going to change in their cells, that they are going to lose their osmoregulatory organelles. Okay, so the second adaptation, that is the physiological adaptations of this parasite, will be parasite can survive in the oxygen deficient intestinal content. Okay, so there will be the less oxygen in this particular situation or the condition where these parasites are going to live after the infection. Okay, so the cysts are resistant to gastric juices. We have seen, we have highlighted it in the last session, that the cysts are resistant, they are capable to get survive in the acidic environment of our gut. Okay, and so that they reach, as soon as they reach in the intestine. Okay, where the environment or the cyst environment changes, and that cyst dissolved in the alkaline medium, so that they are going to form the they are going to form the 8 daughter cells, and that 8 daughter cells are going to convert into the trophozoite stage. Okay, so the third adaptation will be the reproductive adaptations of this particular parasite, that is we are seeing the Entamoeba histolytica, that the rapid binary fission at trophozoite stage. As soon as this parasite is going to convert that quadranucleated stage into the trophozoite stage. Okay, the trophozoite stage is enable capable enough to get get rapid its stage or this get rapid its um, multiplication speed. Okay, so that rapid multiplication in the insisted the stages that to maintain the population of this parasite into the gut. Okay, so outside the body formations of the resistant cyst ensures that the safety of the parasite. Okay, and provides the means to, to reach a new host after the last stage, that will be the quadranucleated stage, and that will be passed through the stools. Okay, so the prophylaxis, that means we can control this if we maintain our hygiene. Okay, the care we should take to get aware or the get rid of these particular problem, that is the get infested by means of the Entamoeba histolytica. So our personal hygiene prophylaxis will be first will be to avoidance of use of raw fruit and vegetables. Okay, we should wash them properly, or we should wash or the we should take their we should take from the uh, such a person that he going to cover the that fruit. Okay, so we are going to second will be the use of boiled drinking water, or rather than use of a filtered drinking water nowadays. Okay, okay, so the protection of a food and a drink from contamination. Okay, we should keep our food, our uh, drinking water away from this contamination zone. Okay, so that these flies and cockroaches are not going to reach to them. Okay, uh, or the we can do the pesticide pest control for the cockroaches also. Okay, obeying the elementary hygiene conditions. Okay, the what are the elementary canal hygiene conditions are there, we should obey the rules of these particular elementary hygiene conditions. Okay, and we should maintain the personal cleanliness. Okay, cleanliness, so that the cockroaches are not going to multiply in our houses. Okay, so the second one will be the community prophylaxis at a large level. What we can do to control these particular third largest parasitic cause. Okay, so the first will be the safe and effective disposals of human excreta. It is most important thing. We should not go to the open places. Okay, and coupled with these sanitary practices like washing hands after the defecation. Okay, when after visiting the our toilets. Okay, we should wash our hand by means of the uh, hand sanitizers or the hand washes. Okay, we should protect of water supplies against fecal contamination. Okay, our municipal corporations are going to supply the drinking water to the our homes. Okay, but they should take care to do not get mixed water with this our um, uh, other wastewater. Okay, so avoidance of a fresh human feces as a fertilizer, this is the as a commodity thing that the human fertilizer or the human feces should use as a fertilizer in the um, farms. Okay, so the health education and the public awareness about the parasite and its mode of transmission should increase. Okay, we should get aware and to get other people aware about this particular parasitic cause. Okay, so the treatment for this particular parasitic cause will be uh, the in divided into three types, that will be First one is the tissue amebicides. Okay, so the amebicides means to which are killing these amebas. Okay, hence known as the amebicides. Okay, so there are the drugs which directly act on the tropozoite stage of the parasite residing inside the tissues. Okay, so the first one will be emetine and dehydroemetine. Okay, that will be referred as the, in short, that will be DHE, that will be emetine and dehydroemetine are the drugs of choice to kill tropozoites. Okay, and resisting inside the intestinal wall, liver, and other metastatic lesions. Okay, so it will be the antibiotic, and the emetine and dehydroemetine, that is DHE, is used in the treatment of this Entamoeba histolytica. So the chloroquine, okay, 4-aminochloroquine, okay, or the chloroquinoline, okay. So it is used specifically for these parasite present in the liver and the lungs. Okay, when the invasion occurs and get these particular parasite are residing in these liver and the lungs. Okay, so the chloroquine and 4-aminochloroquinoline is referred or used in this particular parasitic cause. Okay, so second major dividends will be the luminal amebicides, that means the contact poisons. Okay, so these are the drugs which act when they come in the contact with the tropozoite as well as a cystic forms of Entamoeba histolytica present only in the intestinal lumen. Okay, so that it is why they are also known as the contact amebicides. Okay, that's why these particular drugs are known as the contact amebicides. Okay, so the important luminal amebicides are the diiodohydroquinoline. Okay, it's also referred as the diodoquine. Okay, so indochlorohydroxyquinine. Okay, or it's referred as the clenoquinol. Okay, and hydroxypropaxamine, it's also referred as the mibinol. Chlorobentonide, it's referred as the mantobide. Okay, acetrozone, atrazone, it's also referred as the soratazole. Okay, carbazole, milibis. Okay, histamine, the bismuth iodide, it's also referred as the EBI. Okay, paroxymycin, paromycin, it's also known as the humatin. These are the various drugs are used in the treatment of these particular uh, parasite as a luminal amebicides. Okay, so the third will be the both luminal and the tissue amebicides that are referred as a one time use for these particular uh, luminal amebicides as well as the tissue amebicides. Okay, so the new group of drugs administered orally act on this particular parasite residing in the tissues as well. Okay, so the lumen of these intestine are treated by means of the niridazole group. Okay, that are also known as the ambilihar. Okay, and metronidazole group. Okay, that is also sorry, known by these flagyl, metronidazole, etc. So this was the treatment for used as a in the medicines. Okay, so we are concluding our Entamoeba histolytica, that will be the 5.2 point in this particular fifth chapter. Okay, so we are stopping here. Okay, we will meet on the next session. Thank you.